This is lecture 7, and in this lecture, we will continue to study the B cell development. So we study more the B cell development at the molecular level, how the immunoglobulin loci are arranged in a developing B cell to generate diversity in the variable change, and then after antigen encounter, diversity in the constant region of the heavy change by isotype switching. And so today we will look at how the development occur both at the cellular and molecular level. So what we, again, remember the purpose of B cell development is to generate a great variety of B cell clones that as a single clone, so each clone will recognize one antigen because it will have one type of BCR with a particular amino acid sequence in the variable range of the light change and variable range of the region of the heavy change. That form the antigen binding site, so it will have only one specificity. But as a population of B cells, so all the clones in our body will recognize an unlimited number of antigens. So we said that we have millions of different specificities in our B cells throughout as a whole in our body. And the requirement for the B cell development is not to recognize self-antigens. So although that can happen, and we will see in autoimmunity, the goal is to minimize this, and so the requirement is not to recognize self-antigens. How do B cells develop? And so the B cell development involves sequential generation, assembly, and testing of the newly rearranged BCR. And so ultimately you need to have a B cell that has a functional BCR that is constituted, as we learn, with two identical heavy change, two identical light change, and this BCR, in all cases, will associate with these polypeptides, immunoglobulin beta and immunoglobulin alpha polypeptides. These are polypeptides encodes in germlines. Okay, they don't derive from any rearrangement. Are they always the same? And basically are important because they have a longer intracytoplasmatic tail, and they associate with the constant region of the heavy change, and are the ones that allow the signaling. Okay, so the transmembrane domain of the B cell and the intracytoplasmatic sequence of amino acids in a BCR is very short, and so it doesn't induce signaling, and that's why it associates with these polypeptides, immunoglobulin beta, immunoglobulin alpha, that please do not confuse with the alpha that is the heavy change of the immunoglobulin A, okay? So this is very different. But these polypeptides are important for the signaling. And this is a signaling that involves many kinases. And towards the end of the lecture, we will learn one particular kinase that is key in this signaling. Okay, but ultimately this is the goal. You need a B cell that has a functional BCR expressed in its surface. And so the development can be divided in two phases. The first phase will promote the generation of B cells in the bone marrow, and the phase 2 will promote the elimination of self-reactive B cells that also occurs in the bone marrow. Okay, first, in the first phase, we want to generate functional BCR, and in the phase 2, we want to eliminate any B cell with a BCR that recognizes self-antigens. So an interesting fact about the output of the bone marrow in terms of B cell production is that every day there are 2.5 billion developing lymphocytes start the development into B cells, and then there is induction of proliferation in the cells that have functional BCRs and pre-BCRs, and ultimately there will be 30 billion cells that enter circulation, and most of them will die in few days, but some of them will become longer lived, and they will live for months. Not as long-lived as T cells, as we will contrast in future lectures, but they will live for months. And the other interesting thing that is different with the T cells that are only generated when we have thymus in the young people, the B cells are constantly produced throughout your life. So even if you are very old, a very old person can generate B cells continuously from the bone marrow, new B cells. Okay, let's see what is related to phase 1 here, the development of the B cells. So as I said, this occurs in the bone marrow. We already learned that the B cell derived from the common lymphoid progenitors in the bone marrow. And so this hematopoietic stem cell will differentiate into common lymphoid progenitors. And all throughout the development of the B cells, the stroma cells that exist in the bone marrow will provide important signals and growth factors which promote this B cell development. So here we have some of the stages in the development from the early lymphoid progenitor. Then the first one, once it starts the developmental commitment into B cells, the first stage is called early pro-B cell, then is late pro-B cell, and then is a pre-B cell that can be derived in large pre-B cell and a small pre-B cell, as we will see in the next few slides, and then the immature B cell, and afterward, the mature B cell. But all throughout this development, we have signals from the stroma cells that will dictate and will help the development of these B cells and promote survival in some cases. And so I don't want you to remember all of them because it's too much memorization, but I think it's important that you remember that interleukin-7, IL-7, produced by stroma cells will signal via the IL-7 receptor in developing B cells, and this is essential for allow the survival of these developing B cells. Okay. So now we will learn now that there are different checkpoints throughout the development of the B cells to make sure the BCR, a functional BCR, is on its way. And we will also learn in which sequence the immunoglobulin loci is rearranged to form the functional BCR. And ultimately, we will learn about allelic exclusion and we will learn what that means, okay? So here again are all the stages in the development. So from the stem cell in common lymphoid progenitors, first one is the early pro-B cell, then the late pro-B cell, then the large pre-B cell, the small pre-B cell, the immature B cell, and the mature B cell. And so you can see here there is, in this cartoon, you see in the large pre-B cells, there is a simil BCR that is called pre-BCR, and we will learn more about it. And you see here in the immature B cell, something that you already know, that the mature B cell express IgM, and that the mature B cell express both IgM and IgD. And I just brought up this slide in which we learned that naive immature B cells make only c-mu, and immunoglobulin N is the exclusive isotype that is expressed. And the naive mature B cells now can express mu and delta. And so this is depicted here. Okay, so before we go into how the immunoglobulin loci are rearranged at different stages of development, I just also wanted to bring up to your front lobe the loci, the composition of these loci of the immunoglobulin light chain and immunoglobulin heavy chain. Remember, we have two loci for the immunoglobulin light chain, the lambda light chain locus and the kappa light chain locus, and also the heavy chain locus. And each of them contain in alternative versions of parts of the variable region of the immunoglobulin. For the light chain, alternative Bs, alternative Js. For the heavy chain, alternative Bs, Ds, and Js segments. And we said that for forming the variable region of the light chain, a B and a J needs to come together. For forming the variable region of the heavy chain, a D, J, and V needs to come together. And so how does this occur and which order throughout the development of the T cells? So here you have in the first row, the loci of the immunoglobulin heavy chain. In the second row, the loci of the immunoglobulin light chain. And in the third row is the status of the immunoglobulin in the surface of the developing B cell. And so as all the cells in our body, you can see here stem cells, they have germline constitution in the loci of immunoglobulin heavy and light chain, right? This is the same as in a hepatocyte, right? And only in developing B cells, now these loci will begin to be rearranged. And so in the early pro-B cell, the first stage, the first rearrangement that starts occurring, and this is because there is induction of the expression of the RAG proteins, which are important for these rearrangements of the immunoglobulin, are essential. They will start first the rearrangement of D and J segments. So a D and J from the heavy chain locus will be brought together. After that, the cell mature into a late pro-B cell, and now can start the rearrangement of the VDJ, of a B with the already rearranged DJ, and that is will encode for a rearranged variable region of the heavy chain. And so at this point, they will be a functional checkpoint in this transition. And you can see here the large pre-B cells express a receptor that is called a pre-BCR, that basically is constituted for the a heavy chain that has this variable region that has already been rearranged, and the mu that is the first that is expressed. So it's the heavy chain of the immunoglobulin M. And that's combined with a surrogate light chain that we will learn more about it, to form like a surrogate BCR. Again, it has the correct heavy chain with the variable region that has just been rearranged, and the mu constant region, so a complete heavy chain, and a surrogate light chain. And that's called a pre-BCR, and is expressed in the surface of this developing B cell. And this pre-BCR will be used for the first checkpoint, and if the cells pass this checkpoint, can become a pre-B cell, and that will be expressing this pre-BCR. Okay. If this checkpoint is passed, and so there is a functional pre-BCR, now the a small pre-B cell will start rearrangement of the VJ segment in the light chain locus. And then will this now variable region of the light chain will allow transcription of the light chain. And so now that will combine with the heavy chain. And so in the immature B cells, you will have the mu heavy chain, the lambda or the kappa light chain, and basically this will combine with the combinatorial pairing to express the immunoglobulin M in the surface of these B cells. So in this transition between a small pre-B cell and at the end of the small pre-B cell and the immature B cell is when there is expression of this functional BCR. If this BCR is functional, now the cell can mature into immature B cell that will express that functional BCR, that immunoglobulin M in its surface. Okay, so this is more of what I mentioned about the surrogate light change that basically stabilize the heavy change and allow the heavy change to be expressed as part of the pre-BCR in the surface of the developing B cell, and allows then the testing of the heavy change, that the heavy change was successfully rearranged. Because remember, in these rearrangements of immunoglobulin, there is also incorporation of nucleotides without template, so it may be in frame or may not be in frame, and so it may be productive, this rearrangement, or not. So this is a way, the pre-BCR, in which the successful rearrangement of the heavy change is tested by combining this heavy change, the mu heavy change, that again is the first one to be transcribed, with this surrogate light change. And this surrogate light change is constituted by two proteins, the B pre-B, that is a polypeptide that is mimics the variable change of the light change of the true BCR. And another polypeptide that is called lambda 5 that mimics the constant region of the light change of the true BCR. And so these polypeptides are generated from germline encoded genes. Again, don't be confused. These polypeptides that form the surrogate light change, they are not rearranged, okay? They are just encoded in germline, and they are just serving again to stabilize the constant region of the, the heavy change of the developing B cells that has just been rearranged to check for its functionality. And again, the combination of the heavy change of the immunoglobulin, so two identical heavy change and two identical surrogate light change, form the pre-BCR that allows for the first checkpoint. So now remember that for each of the locus of light change and each of the locus of the heavy change, we have two copies, right? One in each chromosome. And so to make sure there is only one specificity, so there will be only one gene that is rearranged and encodes variable region of the heavy change or variable region of the light change later, the presence of the pre-BCR, when one variable region has been rearranged, when one the locus on the heavy change in one chromosome has been successfully arranged and expressed as a BCR, that sends a signal that indicates the pro-B cell to stop rearrangement of another immunoglobulin gene. Okay, and that's what you want, because otherwise you will have you don't want the two copies of the immunoglobulin heavy change locus to rearrange, because if that's the case, then you will have two versions of variable regions of the heavy change. And if you have two versions of variable regions of the heavy change, you will have two specificities at least. And you don't want that, right? Like the goal is that each clone will have just one specificity. So you need to make sure that when one variable region of the heavy change is rearranged successfully, this will stop rearrangement in the other copy of the immunoglobulin heavy chain gene in the other chromosome, okay? To avoid another version of variable region, to avoid additional specificities in this clone of B cell that is developing. Is that clear? Okay, similarly to that, later in the development, when presence of the BCR in the second checkpoint send a signal to, in this case, it would be in a pre-vessel, in a small pre-vessel, to stop rearrangement of another immunoglobulin light gene. Okay, another means either if it started, always starts with the lambda, and so we want to make sure there's no rearrangement in kappa, and that there's no rearrangement in another copy of the chromosome. So again, because we want to have only one version of the variable region of the light change, to avoid having different specificities within one cell, one clone of B cell. And so this is basically what is called allelic exclusion. So allelic exclusion is first let me tell you, an allele is one of a number of alternative forms of the same gene, right? An allelic exclusion is the mechanisms by which a B cell express only one copy of its two copies of genes. So this, again, allows single specificity per each developing B cell. So the new receptors that are formed, so the pre-BCR in the pre-B-cell, at the end of the pre-B-cell, and the functional BCR at the end of the pre-BCR at the end of the pro-B-cell, and in the transition between pro-B-cell and pre-B-cell, and the functional BCR in the transition between pre-B-cell and immature B-cell, will send signals when they are expressed, will send signals that will result in three outcomes that it's very important for you to remember. The first outcome is the one I just explained in the previous slide, will induce the stop of the immunoglobulin rearrangement, okay? So it will be reduce the expression of the RAG proteins by reducing the transcription of the RAG proteins, or promoting degradation of the RAG protein and other mechanisms. But basically, you want to stop the rearrangement of the other copy of the immunoglobulin heavy chain, or other copies of the immunoglobulin light chain. It will also, and again, that makes sense because we want to have just one specificity in each developing B cell. The second outcome is the induction of proliferation, and that also makes sense because if the pro B cell has generated a functional, the large pre-B cell has a functional pre-BCR, then that's news, and basically there was a lot of energy put on that, and we wanna, this is a success, right? So we wanna make copies of the cell that was successful at generated functional pre-BCR or at generated functional BCR, okay? And so that is mediated via proliferation of the clones that have successfully generated a functional pre-BCR or a functional BCR, okay? So they successfully pass this first checkpoint and this second checkpoint. And the third outcome is the differentiation into the next stage of development. So this clone will proliferate and then differentiate into the next stage of development. In this case, for the pre-BCR, after passing the first checkpoint, will differentiate into a small pre-B cell, and for the immature B cell, will differentiate into the immature B cell. So if this in this transition is the second checkpoint. If there is a correct expression of the BCR, the cell will differentiate into immature B cell. And so this is another way to show there, and also keep in mind that there are multiple chances to rearrange a useful BCR. So here are, again, all the stages: early pro-B cell, late pro-B cell, the pre-B cell, and the immature B cell. And so first is the rearrangement of the heavy change locus. The D and the J are brought together in a random fashion, and then later on, in the late pro-B cells, the V is brought together with D and J in the first chromosome. Okay, if this is successful, if there is a productive joint of these segments, if this is successful, then the cell will pass the first checkpoint with a pre-BCR. And three things will happen. Will inhibit rearrangement on the second chromosome, and this is called allelic exclusion. They will induce proliferation to copy the cells that have successfully rearranged the VDJ, so has successfully expressed a heavy change. And the third one is they will differentiate into the next stage of development, which is the pre-B cell. And in this next step of development, they will start rearrangement of the immunoglobulin light change. And so first is the rearrangement of the kappa gene in the first chromosome. That's how it starts. And so if this is successful, this will combine with the heavy change, the mu heavy change, and will form an immunoglobulin M. And if that's a productive BCR, will pass this pre-B cell, will pass the checkpoint, and three things will happen. Will inhibit any other rearrangement in any other locus of light change or any other chromosome, and will proliferate and will differentiate into an immature B cell that will express the immunoglobulin M in its surface with and the kappa. And so now, in the event that these checkpoints fail, for example, here, if the first checkpoint, the first attempt to generate a mu change in one chromosome fail, and there's no expression of pre-BCR, there's no inhibition of rearrangements of second chromosome. And this developing B cell has a second chance, basically. It can now generate, trying to rearrange successful the heavy change locus in the second chromosome to express a functional PCR. If that's successful, now it's the same. It will pass the first checkpoint with a pre-BCR, will proliferate and differentiate into the next stage of development. In the same way, when a pre-B cell is rearranging the light change, first it will start, as we said, in the kappa gene locus in the first chromosome. If that fails, has another chance to rearrange the kappa gene in the second chromosome. If that is successful, now it can proliferate and differentiate into immature B cell. And will inhibit, by allelic exclusion, any other rearrangement in other immunoglobulin change locus, like the lambda, right? Now, if that is not successful, in the case of the pre-B cell, it has further chances because we have two locus of immunoglobulin light change. And so can start rearrangement of the lambda gene in the first chromosome. If that's successful, now it inhibits rearrangement of lambda genes in the second chromosome and will proliferate and differentiate into an immature B cell that it will express the mu change associated with the lambda light change to express a functional BCR. and if it is not successful, can still have another chance, that is to rearrange the lambda gene on the second chromosome and still generate this functional BCR. And if that's not successful, it will die, okay? So there are multiple chances to rearrange a useful BCR during the B cell development. So each rearrangement has 1 in 3 chances of being successful in frame, okay? And this is because 3 nucleotides will encode from an amino acid. So 1 in 3 chances of being successful or in frame. In the first attempt, if the first attempt is not productive, there is a chance for 1, in the case of the immunoglobulin heavy change, or more, 3 more actually, in the case of the immunoglobulin light change rearrangement, to have further attempts.
I have a question actually. So what exactly is classifying the success of these rearrangements? I know that it says on the slide right here that it's in frame, but is that it?
Excellent. Excellent question, Lindsay. So basically, when is this checkpoint considered okay to pass and give rise to these 3 outcomes, right? This occurs just with the expression of the functional pre-BCR and functional BCR. So it doesn't seem to have, so if they are expressed in the surface, that's enough. And that seems to be enough to induce signaling downstream without any antigen, just the expression of the receptor without seeing any ligand. That's enough to induce signaling downstream that will lead to a stop immunoglobulin rearrangement with allelic exclusion. And this basically at the molecular level means that the levels of RAG proteins will be very reduced, and so will not be able to continue rearranging the other locus of immunoglobulin, other copies of the immunoglobulin heavy chain gene, for example.
Okay, thank you.
Will induce signaling that promote proliferation and will induce signaling that promote further development into the next step.
Okay, thanks. So is there just another molecule that's coming around to check and make sure that everything's expressed? Or is that unknown?
No, it's not known. The truth is not known. It's like the expression is just that these receptors, let's see if I can, when they come together and are expressed, just the expression of these receptors in the surface will be expressed always in association with these polypeptides, the immunoglobulin beta and alpha, and that's enough to promote signaling, or that leads to these 3 outcomes, okay? And it's not known how this is enough to promote the signaling, but that's what is known is that you don't require any ligand, any antigen at this point, okay?
Okay, got it. Thank you so much.
Sure, thank you. Any other questions?
I also have a quick question.
Yes, please.
So for the pre-B cell, when you have the light chains and the kappa light chain locus is, so it's preferentially chosen over the lambda light chain, correct?
Yes, the kappa is first, is the first one that is start being rearranged, and then is the kappa in the second chromosome, and then start, if that is fail, then start the rearrangement of the lambda in the first chromosome, and if that fails, the lambda in the second chromosome. You're correct, man.
And do they have any differences in like the signaling that they have, or is it pretty much the same?
No, any of the 2 will generate a functional BCR, will associate with this mu change that we already know is functional, the rearrangement, because has already been checked in the pre-BCR checkpoint, right? And so if the light change is functional, the mu we already know is functional, and so both of them can form a functional IgM with mu kappa or mu lambda.
Okay.
Any other question? let's go now with the negative selection and receptor editing. So far, we are in a developing B cell that has been successful at generating the functional BCR that has just one specificity because of the mechanism with allelic exclusion. And so we fulfilled this purpose. And that's the phase 1 of the development we mentioned before. But there is a requirement there that the B cell should not recognize the self-antigen. And this is in this phase 2 of the development that also occurs in the bone marrow. And that elimination of self-reactive B cells in the bone marrow occurs in the immature B cell stage if the B cell bounds to cell surface antigen is removed completely from its repertoire. So basically it dies. And so a strong signaling in an immature B cell because it bounds its self-antigens in the surface, for example, of stroma cells, will lead to cell death. And just an interesting fact that you already know, but is interesting to contrast, when a B cell is immature in the bone marrow and see its specific antigen, it will die. And that's an interesting contrast with what happened to a mature B cell that is now in the periphery, that express IgD and IgM and has matured and is circulated in the lymphoid, secondary lymphoid tissues in search of its antigen. Now, when these mature B cells see engage the BCR with an antigen, then the consequence of that engagement is not death, is activation, clonal selection, differentiation into plasma cells that will produce antibodies. And so it's an interesting contrast how the developmental state of the B cell, whether it is immature or whether it is mature, will determine the outcome that of the engaging the BCR with its specific antigen. So the B cells that are specific for their self-antigens, they also have, yes, please.
On the last slide, can you go back to the last
yes.
slide?
Yes.
When you said that the immature B cells, when they experience their antigen, does that have to do with the fact that it's immature versus mature? Or does that have to do with location, like the fact that it's in one place versus the peripheral lymphoid organ?
No, it has to do with the fact that it's immature versus mature. Because if you put them in the same location, if you put them in take them out and you put them in a well, in a Petri dish, and you add the specific antigen, so you compare independent of their location. Now the immature B cell will die with a specific antigen and the mature B cell will be activated.
Got it, thank you.
Great question. Okay, so the B cells that are specific for the self-antigens, they also get a second or third or fourth chance, which is called receptor editing in this case, okay? And so let's say that you have a developing B cell, an immature B cell that now express a functional immunoglobulin M, and so has passed the second checkpoint, and now engaged with a strong ligation antigens, they are specific self-antigens in the cells in the bone marrow. And so that will induce an arrest of B cell development and will allow now these immature B cells to continue, so there will be a regulation of the RAG genes again, and will continue the light chain change rearrangement, okay? And this may, let's say that it rearranged a kappa in the first chromosome, and that leads to a self-reactive BCR. Now when engaged, the self-antigen will arrest the development and will continue the next rearrangement in the second chromosome of kappa. If that's not, if this is still self-reactive, we'll try another continue rearrangement, the lambda gene in the first chromosome, and test it again. If it's still self-reactive, we'll arrest development and will continue rearrangement of lambda gene in the second chromosome, and we'll test it again. If it's still self-reactive, the cell will die, okay? But if not, if it doesn't engage anymore the self-antigens in the bone marrow, now it will continue the differentiation into a mature B cell, and it will express immunoglobulin D and will go into the exit the bone marrow to periphery, to start circulating in the secondary lymphoid organs. So here, a new receptor, when subsequent rearrangement is possible, that a new receptor specificity is now expressed, because the receptor specificity, remember, is given or determined by the variable region of the heavy change and the variable region of the light change. So the variable region of the heavy change will be the same, right? Because you only can continue rearranging the light change, okay? But combined with another variable region in the newly rearranged light change may give this B cell another specificity. And so a new receptor specificity is now expressed in this immature B cell and will be tested again. If the new receptor is still self-reactive, can continue rearrangement other light change locus, and if not anymore to rearrange, the B cell will undergo apoptosis. If the new receptor is no longer self-reactive, the immature B cell will express now IgD and will migrate to the periphery to start circulating in the second lymphoid tissues. Okay. Any question about that? So that's called receptor editing. And basically the mechanism that eliminate a self-reactive B cells in the bone marrow. Sorry, I need to close my window. The mechanism that eliminates self-reactive B cells in the bone marrow is called negative selection. And the mechanisms that allow a generation of a new receptor specificity in the developing B cells that has that is autoreactive is called receptor editing. Any question about that? Okay. Okay, so last we will learn a little bit of an an immunodeficiency that is called X-linked agammaglobulinemia. And so as the name indicates, without gamma globulins, right? And so these are patients that have low levels of gamma globulins or immunoglobulins in their serum. And the reason is because they have a mutation in this kinase that is called Bruton tyrosine kinase, BTK. And so this is a kinase that is encoded in the X chromosome. So this is a disease that affects mostly males because they have only one X chromosome. So if that has the mutation, then they basically will manifest the disease and will have no BTK. And this BTK is necessary for B cell receptor signaling. And so we just learned how the B cell receptor signaling, and so signaling through the immunoglobulin beta and immunoglobulin alpha polypeptides that are associated with the constant region of the heavy change in the pre-BCR and in the BCR are required for B cell development. So this will not occur in these patients, and as a consequence, they will lack B cells. And so these patients have very low levels of immunoglobulins, and also they have a lot of infections with bacteria, particularly encapsulated bacteria. So these have a capsule composed of polysaccharides which protect from the direct attack by the phagocytes. So these bacteria cannot be fought or eliminated by neutrophils because they are protected from the phagocytic and microbicidal activity. They are the phagocytic activity of the neutrophils because they have this capsule, and so they cannot be phagocytosed. And so for being phagocytosed, they require IgG2. And now this IgG2 will opsonize, specific for the bacteria, will opsonize the bacteria, and binding of IgG2 antibodies to the bacterial surface will lead to activation of complements and binding C3b. And this will further opsonize the bacteria and allow now the phagocytosis by neutrophils that then can kill the bacteria. And so the patients that have X-linked gamma globulin, they have enhanced infections with these encapsulated bacteria. Okay, so with that, I would like to summarize what we learned today. So B cells develop in the bone marrow, and for doing so, they need to pass 2 checkpoints. The first checkpoint, we test for a functional heavy chain rearrangement with the formation of the pre-BCR that is formed by the mu change together with the B3b and lambda 5 that form the surrogate light change. And the second checkpoint will be to test the rearrangement of a functional light change, a functional BCR that associated with a mu change that has already been rearranged and checked will find the functional BCR. We also learn about the sequence in which the immunoglobulin gene locus are rearranged. So first is the rearrangement of this D segment of the heavy change with the J segment of the heavy change. Then this DJ is brought together with a B segment in the heavy change, and then late the light change BJ rearrangement. We also learn about what is allelic exclusion, and we learned that the functional pre-BCR or BCR send a signal once it is expressed in the surface, just by being expressed without seeing antigen, will send, if they are successfully expressed because there was a functional rearrangement, then they will send a signal to stop further rearrangement, and that's allelic exclusion. And also this signal will lead to proliferation and differentiation into the next stage of development. We also learned that this allelic exclusion, this inhibition of further gene rearrangement is allowing each B cells to have just one specificity. And you need to understand why is that, right? It's because you only want to have rearranged one variable region for heavy change and one variable region for light change in a particular developing B cell. So negative selection, we learn, is the process by which self-reactive B cells are eliminated from the repertoire of developing B cells. And we also learn about receptor editing, because these self-reactive B cells can undergo subsequent rearrangements of the immunoglobulin light change, okay? They cannot further rearrange the heavy change, it's too late for that, but they can further rearrange the immunoglobulin light change in a process that is called receptor editing. And if after trying a number of rearrangements in the immunoglobulin light change, if the cell is still self-reactive, it will die, again, in this process called negative selection, that is the elimination of self-reactive B cells from the repertoire of the B cells that have been generated in the bone marrow. And last, we learned about X-linked A-gamma globulinemia, which is a deficiency in the BTK or a mutation in the BTK, which is an enzyme, a kinase, glutathione tyrosine kinase, that is encoded in the X chromosome and is important for BCR signaling. So in the patients with this mutation, they lack successful or functional BCR signaling, and so they have no B cells, and they have no or low levels of immunoglobulins, and they have enhanced susceptibility to infections, particularly infections with extracellular bacteria. And so with that, I would like to close this lecture.